You, me, and HIFMB. Stories of Science and the Sea.
Hey everybody, and welcome to the next episode of the HIFMB podcast, and Happy New Year to all of you. I hope you all had a nice Christmas break, and we're getting right into the thick of it again with our podcast, and this time with Jana Massing, who brings to us one of the most complex and theoretical topics we've probably talked about before. So not only are we for the first time talking about bacteria on this channel, we're also for the first time talking about biodiversity theory. The Biodiversity Theory group is one here led by Thilo Groß. Jana is one of Thilo's PhD students, and she brings to us one of her publications, which is currently in resubmission and should be published quite soon, hopefully. And this is on a method that she applied to the bacterial community of the Baltic Sea. The method is called diffusion maps, which is a manifold learning technique. So, without further ado, I give you Jana Massing. Hello and welcome to the next episode of the HIFMB podcast. And today I have Jana Massing with us. Hello.
Hi. Thanks for having me.
Yeah, no worries. From the Theory in Biodiversity group?
Biodiversity Theory, yeah.
Biodiversity Theory. That's the way. Okay.
That's right.
Yeah. So today we want to talk about one of your publications, or your most recent one, I think. I think it's submitted currently.
Yeah, exactly. It's resubmitted,
yeah. Exciting. So this is, the title is a mouthful. It's Quantification of Metabolic Niche Occupancy Dynamics in the Baltic Sea Bacterial Community. So is this your first contact with bacteria? So it's the first time we're talking in the podcast about bacteria, but is it your first?
Yeah, so it kind of started when I started my PhD. So I mean, I studied marine biology, which also involves, of course, marine microbes and bacteria. Yeah. But I think now it's the first time I really got in like close contact and into depth with microbes.
Yeah. Okay. How long have you been working on this study?
Oh, I'm not sure. Maybe like I'm 2 years. Yeah. I'm not sure. Yeah.
Yeah. Okay.
Yeah.
All right.
So because I mean, of course, you submit, right, and then it takes some time, and
exactly,
then you do other projects.
Yeah. Yeah. Submit, resubmit. You get reviews back, and then it stretches for sure. You're a PhD student at HIFMB, but this isn't your first publication.
No, exactly. So it's actually the first project I worked on, but in between we published also a review on generalized modeling.
Yeah, that's one thing. While you're doing your PhD, you're doing many things in parallel, and then other things go by faster than others. Yeah. And what's the status of the study? It's resubmitted and submitted where? In which journal?
M-Systems, that is subjournal of American Society for Microbiology. So, I mean, we used this new method, and we hope that people will use it in the future further on their data.
Yeah, let's get into that. So, the new method that you talk about is called manifold learning, or it's called diffusion maps?
Yeah, so manifold learning is like the category maybe of the method, right?
Okay, right.
And then diffusion maps is the method that we use.
Mm-hmm. The specific method name. So it's a modeling method, not mathematical modeling, not statistical modeling, or
No, it's actually a manifold learning approach, which means, or in this case, it's a dimensionality reduction method.
Oh yes.
So yeah. So maybe I think a lot of people know PCA, which is principal component analysis, right? Okay. So basically, you have high dimensional data, and you're trying to reduce this dimensionality to better understand the system you have.
Okay. And yeah, I imagine bacteria from the Baltic Sea are quite highly dimensional, the data you get on them.
Yes.
So I guess, so in the study, you're kind of, the core, to me, reading the abstract, correct me if I'm wrong, is that you want to link community dynamics with ecosystem functioning for, I guess, one of the first times for bacteria.
Oh, I'm not sure. I think especially if people have looked at like they are equally, all right, or these single species, I think they have done already a great job in analyzing for what the bacteria is important in the system, right? It is in, or if we think about gut bacteria, I think.
Yeah.
But yeah, I think especially in marine systems, it's quite complicated because you have these open systems, right?
Mm-hmm.
Where also you have currents and stuff can get in, stuff can get out. And also these influences, right? So if you, for example, in the Baltic Sea, we have seasonality, meaning all the abiotic parameters are changing, substrates are changing that are available for bacteria.
Mm-hmm.
And
so substrates that they can live and feed on.
Right.
Yeah. Okay.
Mm-hmm.
Right.
And actually, so in our study, we had like over 4,000 different species.
Yeah.
And overall, right, in the marine system, I think until now around 40,000 or over 40,000 bacterial species have been identified. So
okay.
Yeah, this is a lot, right?
That's wild, yeah. And you're trying to kind of show their ecological niche or their yes.
Yeah, we're trying to make one step in that direction, right? That we don't, I mean, when we look at all the species, what we actually want to see, or what is important for us, right, are the ecological functions that are behind this.
Mm-hmm.
Or we also talk sometimes about ecosystem services, right?
Yeah.
So, for example, if we think about bacteria, they are major recyclers. So,
so that's what the ecosystem function we talked about before is.
Yeah, for example.
Yeah.
And
or one of them.
Yeah, right. And also they produce oxygen and stuff like that. And this is also when we think about global change, right? That is something we want to understand what is going on with these communities that are so important to us.
Yeah.
And this is why we kind of need to. So we have their dynamics over time, right? We know, okay, species A is going up, species B is going down, but we have these many species, so we cannot look at every species. One by one, but we actually need something to understand, okay, how is the ecological function changing if the species are varying in their balances, right? Or that time patterns, the dynamics. Yeah.
Absolutely. I imagine just because bacteria are so small and I guess hard to research, are they often overlooked when it comes to ecosystem functions, or?
Yeah, I think so. I mean, also the problem is we kind of just got to know about bacteria, right? Because it's so hard to see them. Also, as you said, we cannot see them, and just with these new technologies, right? Sequencing, for example, we can get some insights on them.
Yeah. So the sequencing is a genetic technique again. So
right. Yeah.
How do you the data set? So you didn't collect them, but in the data set they are you just have genomes, or what is the data of?
The data is actually amplicon sequence variants, which is that you take a very conserved region of in this case, DNA that is encoding the ribosome.
Yeah.
And thereby you can, to a certain degree, identify the species because you know about the species from sequencing before from databases that it has this specific sequence, and then you can say, this is species A, species B, which also means we just detect the species that we already know that they exist. Yeah.
Okay. So I imagine, like we said, it's a very highly dimensional data set that you're dealing with. And what exactly is the manifold learning doing? So the method you talked about, diffusion maps of the manifold learning category, what is it doing? How does it work?
So it kind of detects the variables that explain the variation in the data set. So yeah, basically we have this data set of different bacteria, and then also we have the genes. So for example, they can use glucose or they can do photosynthesis. And with this, it kind of finds the metabolic strategies of the bacteria. So it really says, for example, are you a bacteria that lives associated to a host, right? Then you need you need to be able to do certain things.
Yeah.
And this is...
have certain characteristics.
Right, yeah. And so the method is actually able to detect these different strategies. And... so basically by finding the right variables of the data set. So if we think about high-dimensional data is actually so what is hard about high-dimensional data is that if you compare very distant objects, it's not really clear what variable we should use to compare them. So, for example, if we compare different houses, right, this is kind of an easy task. So you can say, okay, this house has this and this height, for example, or these many doors, or these many people live in this house. But if we think about comparing very distant objects, so for example, if I want to compare a house to a cat or a cat to a song, like what variables do I use, right? What do I ask? Yeah. And this is what the method finds. It finds these major explanatory variables, kind of.
Yeah, great. And then but it doesn't name them for you, right? So it just tells you there's these many explanatory variables. There you go. And then you have to interpret this, right?
Yeah, that's right. Yeah. I mean, at least it tells you which one is the most important, so which one kind of explains the most variation in the data set, which is good, no? Yeah. But yeah, then you need this is why it's, for example, good to have meta information, right? For example, temperature, and also in our case, all of these different genes, so we know what the genes might be doing. And then we can look, of course, in the taxa or in the species, let's say species, that score extreme values in these variables, in these new variables, and see, okay, what is probably the strategy that they represent.
So this is then where the brain of the scientist comes in and interprets what the method gives her or him, and interprets what makes the most sense, right? Or puts, so the method tells you, these are the explanatory variables, and you then interpret what makes the most sense with the metadata you're given.
Yeah, exactly. Or I use these other information, right, to make sense of what I got from the diffusion map.
Yeah, exactly. Yeah. So the method is only as good as the driver in the end.
Yeah, probably.
Yeah. Cool. But it sounds almost magic.
Yeah, I think it is.
Yeah. Where has it been? So I think it's been used in a couple of instances already at our institute, at the very least.
Yeah. So, for example, Alex Ryabov, right? Yeah. He used it also to, in a very cool example, for quantifying functional diversity, right? Because this is actually kind of before, right, you needed to know all these functional traits, and I mean, sometimes we don't even know all the organisms, right?
Yeah, exactly.
And he did this with phytoplankton, which is also, I think, we have not understood everything yet about phytoplankton, and so he kind of from the abundances, so how often they appear in the samples, he used the diffusion map to quantify functional diversity in the samples.
That's wild. Without it being sampled to begin with, like I imagine in your case now the time series goes back to where? Like, was it 2011 or?
Yes. 9 years.
And I imagine they haven't recorded traits or functional traits or anything whatsoever. But then you are able, in hindsight, to put something functional on this time series, which is incredibly useful. Awesome. And what have you found? So what's the main finding of your study?
I think, first of all, maybe that this method works. So because, I mean, for example, what we saw is an important trait was photosynthesis, right? And it also really separated the different bacteria, which makes sense because for doing photosynthesis, you need so many different adaptations, right? If we think, I mean, here when we are outside the ocean, right, only plants basically can do it, I guess. Yeah. And what was also cool, what we saw in the first variable, that it identified the pathogens. So we saw all these very nasty pathogens actually like relatives to Yersinia pestis, right, which caused the Black Death. And we're like, okay, what is this doing in the ocean? But it's actually the bias that this method can identify. So of course, we as humans are much more interested in pathogenic taxa or like bacteria that can cause us harm, basically. And this is why we know much more about them. And so our databases are full of them. And so this method detected this bias. and I hope that we show that we get one step further to understanding these community dynamics, so how the bacteria fluctuate over time, more in terms of, okay, what does it mean for the system, right? If we have different strategies that go up and down.
And then how this also links to ecosystem functioning, which was one of the things we said in the beginning. Cool. So this method works. Where are you going to apply it next?
Yeah, let's see. I think there's a lot to do with this method.
Yeah.
But in this case, actually, I hope that, that the ideal data for us would have been to really get the genomes from the place, right? Because what we now did, we mapped our, so we knew that species A, right? And we mapped this to a database. And I hope with now these new sequencing technologies and so on that we really can get the species from the system and their genome, because there are of course variations between. Also, bacteria are very complicated, so there's also variation in between species, and that would be great to then go even deeper, right? And see, okay, what is for example special to the Baltic Sea microbiome.
Yeah. Okay. And just maybe for the listener to make it more visual, like how do you apply this method? I read in your CV that you are versed in the programming language Julia. Programming languages like R, Julia, so R, the letter, are all languages that you type into your computer. Basically, you type a certain phrase into your computer, and your computer understands it. And then makes a statistical thing out of it, does an action out of it. Julia, I think, is one of the ones that I've heard most associated with mathematicians and seems to be the most cutting edge of them currently. How long have you spent learning this language and using it?
Yeah, so actually I had not done a lot of programming before my PhD, so I basically started off with Julia, which was kind of good. I mean, I had done some things in R. I think a lot of biologists use R, but I love Julia because it is just you can write things like you think them, right? So I think there are these programming languages where you always need to define, for example, different types and so on, so that it can only. For example, if it needs to calculate something, it needs to know what is the type, the data type. So, for example, like an integer or a float or whatever, right? Yeah. And Julia is very flexible and just tries to do whatever you put in. So this is pretty cool, and it's very efficient. So it's as fast as C, I think, and C is very hard to program. So it kind of is easy to write, but it's also fast.
Okay. Yeah. And just for visualization, so it looks, at least R looks a little bit like when you open the console on your computer and then you type something in. Does Julia look the same? It's just a console, or?
I think it looks very similar to R.
Okay. All right. Okay. That's how the magic works. That's how the magic happens. Okay. And since we're already talking about your CV, let's get into that into you as an academic. So your academic career kind of started in 2014. In Münster, Germany, and you started as a bachelor in bioscience. And I was wondering how does that compare to biology? What's the difference?
Yeah, I think it's just more chemistry and physics.
Right. Okay.
I think so. I was always already interested in marine biology, but I thought I start out a little more broad because I was not so sure. And I think it was actually good that to have also this basic understanding, right, of chemistry and physics.
Right.
But it was, of course, also tough coming from school, right, where I feel like you don't really learn so much in depth. Yeah, but it was a good experience. It was I learned a lot of different stuff, I think, and also got me interested in so many different things.
Yeah. Did you enjoy it?
Yes, mostly.
Yeah. And you did it at I just saw that you did it at the Institute of Evolution and Biodiversity. Did you know that you're going to end up working with biodiversity?
No, I did not.
Okay. And then you moved to Bremen, where I did my master's as well. I did the same master's study 3-4 years before you. And so it's the Masters of Marine Biology at the University of Bremen.
Right.
And you worked specifically with the upwelling Humboldt current in Peru in your thesis. Did you get to go to Peru?
Yes, I went to kind of, right. I was on the ocean in front of Peru. I was never in Peru.
Oh, right, you didn't.
So
okay.
no, actually, like we flew to Panama and we went onto the research vessel in Panama, crossed the Panama Canal, and then went to the Humboldt current system, and then to Chile. So
was it a German research vessel or?
Yeah.
Yeah. Which one?
The Merian.
Oh, okay. Don't know that one. Okay. I thought maybe it's one like the Polarstern or Sonne or.
No.
Okay. And so you then work with food web structure and trophic interactions, analyzing the entire food web, or
we tried.
Yeah. Okay.
So we used stable isotope and fatty acid analysis to look into the food web structure, and that means you just basically need a little piece of every organism, right? So everything we could get our hands on, we kind of got ourselves a piece and analyzed. And with this, you can kind of see on which position the organisms are in the food web.
A little bit like eDNA, like environmental DNA. We talked about it in, I think, episode 3 or something.
I think it's a bit different because it's telling you more. So basically, you have like this the basis of the food web, like for example phytoplankton, which is eaten then by, for example, copepods and so on. And this level, like if you are on the basic level or one level higher or even one level higher, or if you are like a top predator, this you can kind of approximately determine with this. But I guess with eDNA you can get even more insights.
Yeah, right, okay. And I just saw in your publications record as well that you last year actually published the publication that goes along with that cruise.
Right. Yeah.
Did you get to write it during your PhD or?
Yeah,
kind of
it was kind of stressful, so I had to finish it in the first year of my PhD. Also had to learn a lot of new stuff, of course. Yeah.
Yeah. Okay.
But I still wanted to. So I thought we had some nice insights, and I wanted to write the paper.
Yeah. Nice. Yeah, and I really like the start of the title, "Toward a Solution of the Peruvian Puzzle," and I was wondering what's the puzzle.
So the Humboldt current system is one of the 4 eastern boundary upwelling systems.
Okay.
Which is kind of so it's special because due to specific currents, ocean currents, nutrients are coming up, and this means it's a very productive system because it has so much nutrients, kind of energy. And the other systems have the same phenomena with the currents, and also actually the same amount of primary production. But the Peruvian system is 5 to 8 times higher in fisheries yield, and we were wondering, like, wow, why is this happening? And as we know, the prime production is the same, right? And kind of the output is the same. So the fish, it's more, but the kind of output.
Yeah.
We thought that it must be in the structure, right? So in between, and this was actually the case. So what we found was that it's a much simpler system. So basically, you channel all the energy towards the fish.
Yeah. Okay. Wow. And then, so this was your master's thesis being finished. Then you went into the PhD at HFMB. How did you, did the working on the food web structure and trophic interactions, did that kind of spark your interest in these theory, into the theory thinking part? And then you applied for the position, or?
Yeah, I think there were 2 main reasons for this transition. Okay. So first, for my master's thesis, I had a lot of data, right? As I said, we collected pieces of everything we could get our hands on.
Sorry. How long was the cruise in Peru?
6 weeks.
Oh, right. Okay, yeah. A lot of data to collect.
Yeah. Yeah, but then kind of I felt like, okay, we are missing tools to really analyze the system, right? I mean, we of course plotted the data and stuff like that and did some statistical analysis, but I felt like, okay, this is so much data, and I feel like I have to do more with this, right? We did all this effort, and now I really want to understand what is going on in the system, and I want to have tools to analyze the data. And this is actually when I read the PhD, like the position, the job posting of this PhD position I'm now in, I was like, okay, this is what I want to, what I always asked myself, right? If there are tools for this.
Mm-hmm.
And the second thing was that I was in this winter school on ocean governance, and our director, our now director, right? So Helmut was there, and he was talking about biodiversity.
Yeah.
And before that, I of course knew about biodiversity, but he gave such an interesting talk that I got interested in the HFMB.
Mm-hmm.
And in this biodiversity theory and modeling stuff.
But because you mentioned the winter school for ocean governance, are you also interested, or were you also interested in ocean governance? And so this is quite a lot of fields that you're tapping into.
Yeah, there are many things I'm interested in.
Yeah.
I think it's very hard to decide.
Yeah, okay, right.
No, but I think also everything so it was just an opportunity that came up because I was working at the ZMT at that point.
Mm-hmm. Okay.
And they had the ocean.
The Leibniz, the ZMT is the Leibniz Center for Tropical Marine Research by now, right? Or
did it change? I don't know.
I think it was Tropical Marine Ecology at one point,
Okay.
and now it's research. I think.
Could be. I don't know.
Okay. All right. Sorry, I interrupted you.
Oh no, I just wanted to say that I think it's just nice to always get these new perspectives from whatever side. Yeah. And but I don't know where I will end up.
How long was the winter school?
I think it was only like 2 weeks.
Okay. All right. Do you see yourself going back to ocean governance or going into it more?
I'm not sure because I haven't done anything in that direction, I would say.
Right.
But I think it's very important, and maybe I would like to collaborate with those people if there is anything I could do, right?
Yeah, absolutely. Yeah, with the governance group here, for sure there's going to be opportunities.
Yeah, for example.
Yeah. Sweet. Okay. And now since we're kind of already talking about your other endeavors that you've tapped into, you also you're one of the hosts of Wissenschaft fürs Wohnzimmer, a series that was it always on YouTube?
I think so. Yeah.
Yeah. Okay. Mm-hmm. So it's a series that is on YouTube interviewing various scientists from. Is it only Alfred Wegener Institute and?
No, it's all kind of institutions.
Oh, right, okay. And I got interviewed as well at some point, I think in episode, 100 and something, maybe.
Yeah, I guess.
Probably. Yeah. How did that start? Or how did it come up? Were you involved in the beginning?
No, I was not involved in the beginning, but it kind of started, I think, with Corona.
Yeah. Okay.
So maybe one good thing from this. And I think, before, for example, there was this Science Goes Public, right, where scientists went into pubs and just talked about their science there. And then during Corona, I think the people that started that were like, okay, now we cannot do this stuff anymore, but science communication is so important, especially with all the issues we have right now. And that's why they started to just say, okay, why don't we have this on YouTube? And
Sweet. Are you one of 3 hosts, right? Or
no, there are more people involved.
Ah, okay, right.
Yeah, I think they are like, I don't know, maybe like 8 to 10 regular people.
Yeah.
Yeah.
Sweet. Do you see yourself in doing more science communication stuff, or?
So at the moment I don't have so much time, right? Because I'm kind of finishing my PhD.
Yeah, that's right. Yeah.
But I think it's really important, and I also really enjoy it. And I think sometimes you can get even these really cool insights from people that have nothing to do with your science, right?
Yeah.
Which is also so cool. I think the questions asked are sometimes so good. I hope definitely to continue somehow working or somehow doing science communication.
Yeah, wonderful. Okay, yeah, since you mentioned you're kind of finishing, how much time do you have left on your PhD?
So my contract ends in October.
Okay, all right. Do you have anything on the horizon, or where would you like to go next?
Yeah, that is what I should now figure out. And I'm trying to figure out.
Okay, yeah, no pressure. Sorry.
Yeah, no, that's fine. But I have so many things that I find interesting, right? And but I think I definitely want to continue to work interdisciplinary, because this is what I enjoyed most now in my PhD, right? Working with so many different people and learning like all these new tools, which is also it's also hard, but it also is I think so cool. So it gives you new perspectives on the problems. Like, for example, before, right, I was like, with this practical work, it's more like you try to go and you try to measure the system as specific or as in detail as you can. Yeah. And with modeling, I had to like completely throw this overboard and be like you cannot. You cannot. So if you represent the system as complicated as it is, you will not get any new insights, right? So you need to simplify. But as a biologist, I was like, but this is missing in the model. Yeah, but we need to simplify, and then we can hopefully get some insights from this, right? Yeah. Yeah.
Wow. Okay. But I mean, it looks like since you've kind of tapped into so many different fields in such a short amount of time, and you've kind of done a very good job of it. Like from my perspective, I think you're gonna do just fine in your wherever you go next. Is it gonna be? But it sounds like it's gonna be science. You're happy with science?
I'm excited about science, but I guess I will also. I mean, like I said, with the science communication, I also like, and also sometimes I think about working in an NGO, right? So I'm not sure yet.
Cool.
Get old.
Exciting stuff ahead for Jana.
Let's hope so.
Cool. Okay, that's we're already at the 33-minute mark, and I think that's all from my side. Is there anything that you want to mention that we haven't mentioned, or that you think we've missed? Anything left?
No, thanks. Okay. Thanks a lot. I think the only thing that I sometimes think that when I would start my PhD again, I would like me to have more courage. So I think if you start especially diving into completely new fields, right? I think at first you're like, oh my God, I don't know if I can do this. Yeah. But I would say you can do it.
That's perfect words to end on. Thank you very much.
Thank you.
Okay, cheers. Bye-bye, everyone.
Want to dive deeper? Surf over to hifmb.de or follow us on Twitter at hifmb_OL.